Chapter 3. Reading the Code. Bringing DNA to Life. Long before Oswald Avery's experiments put DNA in the spotlight as the transforming principle, geneticists were trying to understand just how the hereditary material, whatever it might be, was able to influence the characteristics of a particular organism. How did Mendel's factors affect the form of peas, making them either wrinkled or round? The first clue came around the turn of the century, just after the rediscovery of Mendel's work. Archibald Garrod, an English physician whose slow progress through medical school and singular lack of a bedside manner had ensured him a career in research rather than patient care at St. Bartholomew's Hospital in London, was interested in a group of rare diseases of which a common marked symptom was strangely colored urine. One of these diseases, alkaptonuria, has been dubbed black diaper syndrome because those afflicted with it pass urine that turns black on exposure to air. Despite this alarming symptom, the disease is usually not lethal, though it can lead in later life to an arthritis-like condition as the black urine pigments accumulate in the joints and spine. Contemporary science attributed the blackening to a substance produced by bacteria living in the gut. But Garrod argued that the appearance of black urine in newborns, whose guts lack bacterial colonies, implied that the substance was produced by the body itself. He inferred that it was the product of a flaw in the body's chemical machinery, an error in metabolism, in his words, suggesting there might be a critical glitch in some biochemical pathway. Garrod further observed that alkaptonuria, though very rare in the population as a whole, occurred more frequently among children of marriages between blood relatives. In 1902, he was able to explain the phenomenon in terms of Mendel's newly rediscovered laws. Here was the pattern of inheritance to be expected of a rare recessive gene. Two first cousins, say, have both received a copy of the alkaptonuria gene from the same grandparent, creating a 1 in 4 chance that their union will produce a child homozygous for the gene, that is, a child with 2 copies of the recessive gene, who will therefore develop alkaptonuria. Combining his biochemical and genetic analyses, Garrod concluded that alkaptonuria is an inborn error in metabolism. Though nobody really appreciated it at the time, Garrod was thus the first to make the causal connection between genes and their physiological effect. Genes in some way governed metabolic processes, and an error in a gene, a mutation, could result in a defective metabolic pathway. The next significant step would not occur until 1941, when George Beadle and Ed Tatum published their study of induced mutations in a tropical bread mold. Beadle had grown up outside Wahoo, Nebraska, and would have taken over the family farm had a high school science teacher not encouraged him to consider an alternative career. Through the 30s, first at Caltech in association with T.H. Morgan of fruit fly fame, and then at the Institut de Biologie Physico-Chimique in Paris, Beadle had applied himself to discovering how genes work their magic in affecting, for example, eye color in fruit flies. Upon his arrival at Stanford University in 1937, he recruited Tatum, who joined the effort against the advice of his academic advisors. Ed Tatum had been both an undergraduate and graduate student at the University of Wisconsin, doing studies of bacteria that lived in milk, of which there was no shortage in the cheese state. Though the job with Beadle might be intellectually challenging, Tatum's Wisconsin professors counseled in favor of the financial security to be found in a career with the dairy industry. Fortunately for science, Tatum chose Beadle over butter. Beadle and Tatum came to realize that fruit flies were too complex for the kind of research at hand. Finding the effect of a single mutation in an animal as complicated as Drosophila would be like looking for a needle in a haystack. They chose instead to work with an altogether simpler species, Neurospora crassa, the orange-red mold that grows on bread in tropical countries. The plan was simple. Subject the mold to X-rays to cause mutations, just as Muller had done with fruit flies, and then try to determine the impact of the resulting mutations on the fungi. They would track the effects of the mutations in this way. Normal, i.e. unmutated Neurospora, it was known, could survive on a so-called minimal culture medium. On this basic diet, they could evidently synthesize biochemically all the larger molecules they required to live, constructing them from the simpler ones in the nutrient medium. Beadle and Tatum theorized that a mutation that knocked out any of those synthetic pathways would result in the irradiated mold strain being unable to grow on minimal medium. That same strain should, however, still manage to thrive on a complete medium, one containing all the molecules necessary for life, like amino acids and vitamins. In other words, the mutation preventing the synthesis of a key nutrient would be rendered harmless if the nutrient were available directly from the culture medium. Beadle and Tatum irradiated some 5,000 specimens, then set about testing each one to see whether it could survive on minimal medium.
This book is continued on Disc